Hello and welcome back to the channel everyone. Follow us, subscribe to the channel and hit the bell icon for all the updates. Hello everyone, welcome back to channel Pharmacology Simplified. In today's video, I would be discussing about the Parkinson's disease. I would be dividing this video into I would be covering this topic into two videos. First part would deal with the physiology and the pathological aspects of Parkinson's disease, and the second one would cover the pharmacological agents that are used for the management of Parkinson's disease. So when we talk about Parkinson's disease, it is a disease, we can say it is a neurodegenerative disease, and it is a progressive neurodegenerative disease. Progressive means that this disease, if not managed on time, it gets worsened over a period of time. So it is a progressive neurodegenerative disease that primarily affects the body posture and the body movement. So in this video, I would be covering, as I already said, the physiological pathways, the pathophysiology of Parkinson's disease and the pharmacological management. This would be covered in part 2. So when we talk about Parkinson's disease, the very first thing that we have to understand is the neurotransmission, the process of neurotransmission. If you have any doubts, I am providing the link to the neurotransmission video here. You can go and have a look how neurotransmission occurs and make yourself familiar with the term EPSP and IPSP. So this time in Parkinson's disease, see, when we talk about Parkinson's disease, few things that we have to take care of. The very first thing that we have to take care is the basal nuclei. This is in some texts, you would also find it as written as basal ganglia, as earlier it was called basal ganglia, but now it has been renamed to basal nuclei, because nuclei are the cluster of cell bodies inside the CNS, while ganglia are referred to as the cluster of cell bodies or the collection of cell bodies outside the CNS. So in the basal nuclei, before that we have to understand that how a movement is initiated in our body, a voluntary movement is initiated, and how this gets disrupted during Parkinson's disease. So whenever a movement is initiated in the body, this, for example, I have to write something. So first I have to think about it, like now I have to write something. So that thought has to originate in my cerebral cortex, right? So when the thought will arise in the cerebral cortex, this cerebral cortex sends signal to the basal nuclei, right? The first signal, initial signal from the cortex, when I have thought about it, so this cortex will send signal to the basal nuclei, and there are several components of the basal nuclei, but the primarily that we would be discussing here are these caudate nucleus. This does not play a very major role in Parkinson's disease. We would be discussing about putamen, globus pallidus internal and external, and subthalamic nuclei, right? So this cortex sends signal to basal nuclei. These all so the interwiring of the various nuclei in the basal nuclei, what they do, they rectify and they initiate a smoother movement, and they send the signal to the thalamus. From the thalamus comprises of several individual nuclei. It's a cluster of nuclei. I'm not going into the detail of those. Now this thalamus sends the signal back to the cortex, right? It sends signal back to the cerebral cortex, and from the cerebral cortex these signals go to the spinal cord, and from the spinal cord these signals, they are propagated to the skeletal muscles. So now first we have to understand the physiological pathways, how these physiological pathways they function, and how movements are initiated, how movements are coordinated, and how movements are smoothened, and how during Parkinson's, how the destruction of dopaminergic neurons in the brain, because Parkinson's disease is developed because there is destruction of dopaminergic neurons in substantia nigra, and substantia nigra, it comprises of two components, substantia nigra pars compacta and substantia nigra pars reticulata. Substantia nigra is a part of the midbrain. It is entirely separate from this basal nuclei, but all the dopaminergic activities, they are taking place through the substantia nigra only, right? So now we will see individually that what are all these pathways. So now the arrangement that we are seeing here is comprising of this. This is basal ganglia. It has different nuclei. This one is called putamen. This is globus pallidus externa. This is globus pallidus interna. This whole putamen, globus pallidus externa and interna, they are collectively called as lentiform nucleus, and there is one separate nucleus called as caudate nucleus. It is tail shaped, and lentiform nucleus and caudate nucleus together they are called as stratum. Along with this, there is also thalamus. Thalamus plays a role in smoothing of the movement and coordination of the movement. This is subthalamic nuclei. This acts as a relay station for impulses from globus, from this lentiform nucleus to the thalamus. There are two pathways, direct and indirect. I would be discussing them separately. And this is substantia nigra. Substantia nigra is the place where all the dopaminergic neurons are present, and it has two parts: substantia nigra pars compacta and substantia nigra pars reticulata. Pars compacta is called so because it has very dense cluster of dopaminergic neurons, right? So I would say you write all these names because I would need to clear them off before I can draw the neuronal pathways. And yes, needless to mention, this is the cortex. This is the cortex. So you please write down these names. Please mention the nomenclature. So now if we see here, what I am showing here are the two pathways, right? Two pathways called as the direct and the indirect pathways. So first, as I just mentioned, like for initiating any movement, the thought has to arise in the cortex. So this is the cortex. So from here the thought will arise, and after the thought arises, the thought first goes to the basal ganglia, then it goes to the thalamus, then it again comes back to the cortex, and then it goes to the spinal cord. So now if we see, first we will see this neuron. In the blue I have shown glutaminergic neurons, which means they are excitatory neurons, and in the red I have shown the GABAergic neuron, which are inhibitory in nature. So whenever there is a rising of a thought of initiating a movement, the very first thing is that this movement will be initiated from the cortex, and a glutaminergic neuron will send this signal to the putamen. Right? So here glutamate, what is the nature of glutamate? It is stimulatory, which means that there will be release of GABA at this point. There will be release of glutamate at this point, and it will stimulate this neuron. And this neuron is GABAergic, right? This neuron releases GABA, and this neuron, when it releases GABA, it will generate IPSP here, right? There will be generation of inhibitory postsynaptic potential here, and this GABA will now inhibit this neuron. And when this neuron will be inhibited, then there will be no release of GABA here, or there will be generation of EPSP at this neuron. This neuron is a glutaminergic neuron, which means it is excitatory in nature, and then it goes and sends signals to the areas of the cerebral cortex, where it will send the signals to the spinal tract, and then there will be contraction of the skeletal muscles, right? Any voluntary contract happens in this way, right? The other pathway, it is slightly different from the previous one. Here what happens, the signal, it will arise, the thought will arise in the cortex only, and from the cortex the signal will come to the putamen as it was coming into the direct pathway. Now when it comes into the putamen, now there is a slightly difference in the wirings. So now there will be release of glutamate, which means there will be excitation of this neuron. This neuron is a GABAergic neuron, which means this will release GABA here, and this will lead to inhibition. When this neuron is excited, as a result of which this neuron will be inhibited because there is release of GABA. This neuron is inhibited, and then there will be no release of GABA, or very less release of GABA. And now what will happen? This is a glutaminergic neuron, so this neuron will be stimulated. When this neuron is stimulated, there is release of GABA, there is release of glutamate, and when glutamate is released, there will be release of glutamate. It will stimulate this neuron. This neuron will get stimulated, and then it will release GABA here. When it will release GABA, there will be inhibition of this neuron, right? So there will be no excitatory impulses going to the brain, right? So now what is the importance, or what is the significance of both the pathways, right? The direct and indirect pathways. Why it is important to have two pathways and not just one pathway? For example, if you want to flex one muscle in your body, right? If you want to flex one muscle in the body, then it is necessary that we have the agonistic muscles and the antagonistic muscles. You must be aware of that. If not, I am explaining it in very brief. For example, this is your humerus, and these are your radius and ulna, right? For generating this movement, right? For generating this movement, it is necessary that your biceps muscle has to contract, and along with that your triceps muscle, right? Your triceps muscle, they have to relax, right? So there has to be the simultaneous stimulation and inhibition of two different muscles. So what is happening here? Your biceps muscles, they are getting contracted. At the same time, these triceps muscles, they are getting relaxed. So here we are watching that this is a stimulatory input and this is a inhibitory input through the on the biceps and triceps muscles. So this happens with every muscle in the body. For example, if I have to tilt my neck to the left, so these muscles, they have to contract and they have to relax at the same time. I have to put one foot forward, then one of the muscles who are taking the foot to the back, they have to contract, and the other muscle they have to relax. So this will always happen. So we need two pathways. One is the direct indirect pathway, agonistic and the antagonistic actions on the muscles. So now what is so this is it. Now we see like what is dopamine doing, what is the role of dopamine. We know that dopamine is associated with our behavior and the reward system in the brain, right? Because if we get habit of doing a particular thing, then there is release of dopamine, we feel good about it. For example, people are addicted to their mobile phones, and what they do, that every 5 minutes, 10 minutes or even lesser time than that, what they do, they look at their mobile screens, and what happens during that time? There is release of dopamine in the brain. So this becomes a habit, continuous habit. So in context of Parkinson's disease, this is not related to the mobile phones, but this has to do something with the muscle movements, right? So when the muscles move, what happens? We have seen like people who are playing sports every day, what happens that they become extremely good at a particular type of movement. For example, if we take a bowler, right, in cricket who throws a ball, his movement becomes so much coordinated that out of 100 times, he approximately 98-99% he has the accuracy rate, right? So what has happened? Like dopamine has conditioned his brain, his muscles, they have learned, the muscles have memorized every movement, like this movement has to happen, this muscle has to contract, this muscle has to relax. So this is the role of dopamine, that it is reinforcing the muscle contraction in the brain, right, in the body, right? So this is substantia nigra pars compacta. So it goes and synapses with the with the neurons of both the direct and the indirect pathway. The interesting thing here is that here we have two different types of dopaminergic receptor. These are D1 and D2. D1 receptors are present in the direct pathway and D2 receptors are present on the neurons of the indirect pathway, right? And one important thing also that I have to tell, that D1 is stimulatory and D2 is inhibitory, right? So D1 receptor is stimulatory and D2 receptor is inhibitory in nature. So whenever there is binding of dopamine on D1 receptor, it will lead to the stimulation of the neuron on which this D1 receptor is present, and vice versa will happen in the case of binding of dopamine to the D2 receptor, right? So now we see that on this neuron on the, let me draw this by green. So here we have our, on this neuron, on this GABAergic neuron, we have our D1 receptor, and on this we have our D2 receptors. Sorry. So what happens when this is, see, I told that dopamine is associated with habits. So whenever there is a thought of initiating a muscle movement, this dopamine will be released and it will bind on both the receptors, D1 and D2 receptors. The D1 pathway is not the dominant pathway, but the D2 pathway is primarily the dominant pathway in the muscle contraction process. So now we have to see this very clearly. This glutamate was released, this was binding onto this neuron and it was causing the stimulation of this. So now when dopamine will bind to this D1 receptor, D1 is excitatory, so there will be more strong influence, right? So the action potential generated will be more strong than the previous one. So this happens when we continuously repeat a movement. So under the influence of D1, there will be excitation of this neuron, and this excitation will lead to more release of GABA. So this neuron will be more strongly inhibited, right? Now this neuron is more strongly inhibited, and there will be very less release of very less release of GABA, and there will be less inhibition, or this neuron will be freed from the inhibition. Now this neuron will undergo EPSP, excitatory postsynaptic potential, and now the impulses that are going to this area will be much stronger, right? So any muscle that was contracting, right, any muscle that I was that the individual was contracting will become more powerful. That muscle will contract even more strongly, right? Now the second thing is, this dopamine will bind to D2 receptor as well. And when it binds to D2 receptor, what I told you, that D2 receptor is inhibitory in nature. So when dopamine will bind on this D2 in this D2 receptor, it will inhibit this neuron, right? So this is getting opposite. So now this glutamate was stimulating this neuron, but what this D2 will do, this will inhibit this neuron, right? And when this neuron will be inhibited, there will be no release of GABA. And when this GABA is not released, there will be stimulation of this neuron. This neuron will be stimulated. And when this neuron will be stimulated, there will be release of GABA here. And when GABA is released here, this neuron will be inhibited, right? So what was happening in this case, now it is not happening anymore. So now this is inhibited, so there will be less release of glutamate here, right? Less release of glutamate, which means this neuron will not be excited. This neuron will undergo inhibition, and when this neuron will undergo inhibition, there will be very less or no release of GABA, which means this neuron that was earlier inhibited, now this neuron will be showing very strong, will send very strong signals to the brain for the muscle contraction. So now this seems a bit confusing, but it is not like that. See, there are no separate boxes that are inserted in our brain, right? There are areas that are innervated by all sorts of neurons. So if we are planning on doing one activity, it is not an independent activity. Several other processes are also altered because of the first activity that I have started to take. So what was happening here? These neurons were converging, and for example, if I say from this side, right? From this side, what was happening? There was impulses going down. I have crossed it because there is deconcentration of the pyramids in the medulla oblongata. So right side controls the left and left controls the right, right? So this is going to the spinal cord, right? This is going to the spinal cord, and there will be contraction of the muscles. So when, for example, if I am trying to contract my biceps, so there has to be occurrence of two events simultaneously. This has to contract and this has to relax. These muscles, they have to relax. Sorry. So now what will happen if we see that both some neurons, they will be the inhibition of the muscles, it will become strong, and those muscles that are supposed to be contracted, it will become strong. So what dopamine has done, that dopamine has feeded, right? Dopamine has strengthened the impulses, both the contraction and relaxation. The cycle of the muscle contraction, it has been strengthened by the dopamine feed, right? Now, the actual scenario, the actual chaos starts when, due to one or the other reason, there is destruction of dopaminergic neurons in the substantia nigra, right? So when there is destruction of these neurons in substantia nigra, what happens? When there is no dopamine, when there is no or very little dopamine present in the substantia nigra, what happens? This dopamine is not available here to bind, right? And when this dopamine is not available here to bind, then this stimulation falls down, right? This stimulation falls down, and the signals, they become very weak. So what pathways, what was happening earlier as a smooth, strong, coordinated movement, now what becomes? It becomes a slow, jerky movement, right? Because the activities are not coordinated. Because the impulses that were being fed from this side, right? The impulses that were being fed from this side, now they are not being fed. So now what is happening? These muscle movement, they lose their coordination, right? Right? These muscles, they lose their coordination, and at some time some muscles are contracting and the other muscles are relaxing. So there is incoordinated contraction of the muscles, and if we see these symptoms, they are called as tremors, right? So this is how during Parkinson's an individual develops tremors, right? So this is the whole process, how how the muscle contraction, it occurs in an individual, how dopamine feeds it, and how the destruction of neurons in the substantia nigra leads to the irregular movement of the body, right? And not only this, the patients that are suffering from Parkinson's disease, they not only find it difficult to maintain coordinated movements, they also find it very difficult to initiate a movement, and they also find it very difficult to inhibit or to stop the movement they are doing. For example, if they are moving their hand like this, and if I say I have to stop my hand right here, I can do it like this. But a Parkinson patient, first he or she will have excessive difficulty in initiating the movement, and the movement will not be as smooth as this. It will be like a jerky movement, right? The movement will go something like this, and if I say that you stop right now, they cannot stop here. They will go at some point ahead, right? They will go beyond this point because their muscle activities are not that well coordinated, right? So this is how it is happening. One more thing that I'm not drawing here, but there are neurons of acetylcholine as well, right? There are some cholinergic neurons as well, and what these cholinergic neurons do, they oppose the actions of dopamine, right? So in short, we can understand this, that during Parkinson's, for example, if I say acetylcholine and dopamine, right, during a physiological state they are in this position, but since there is destruction of dopaminergic neurons in the brain, in the substantia nigra, what happens? This becomes slightly disoriented. So the actions of acetylcholine, they become more, right? And these excessive levels of acetylcholine in the brain, they actually lead to tremors and the rigidity in the body posture, right? The body posture also gets disrupted, right? So with this, I would like to put an end to this part of the video, and in the second part, I would be discussing about the pharmacological management of the drugs that are used for managing Parkinson's disease. So if you have been able to understand anything out of this, and if this seems helpful to you, please make sure to subscribe to the channel, share the video as much as possible, and happy learning to all of us. Thank you very much.